Good morning. Today we will be introducing our iGEM project. Before the four main chapter is our brief intro. The immune system is a powerful tool for killing tumor cells, and immunotherapy has also been a landmark in the history of the human fight against cancer. We designed a tumor-specific gene nanomedicine that can target a wide range of tumor cells, which is expressed only in tumor cells and enhances T cells' immune responses. Building upon the established in vitro stimulation of T cells with antibodies against CD3 and CD86, and the pivotal role of IL-2 in the immune response, we associate the possibility of developing a tumor-specific genetic nanodrug using a specific promoter. This drug could specifically drive tumor cells to express anti-CD3 as scFv and anti-CD86 as scFv, while mobilizing tumor cells to secrete IL-2, a key cytokine to activate T cells and enhance immunotherapy. Upon successful transform of tumor cells, we anticipate a significant increase in the immunotherapy effect. Our presentation contained 4 parts: project, safety, human practice, and team. Through this presentation, we hope to give you a better understanding of our research progress in cancer immunotherapy and the collaborative efforts of our team. Now let's come into the project part. And here is the 4 sections: description, contribution, engineering, and experiments. Section 1, description. Cancer remains a significant medical challenge due to its high mortality, short survival periods, and low cure rates. It arises from genetic mutations that lead to uncontrolled cell growth, influenced by factors such as genetic predisposition, carcinogen exposure, and chronic inflammation. In 2020, cancer caused around 10 million deaths globally, according to WHO, and survival rates, particularly for late-stage cancers, remain low. Despite ongoing research, cancer remains a major unsolved global health issue, highlighting the need for continued innovation in treatment. The immune system is a powerful tool for fighting cancer, and while immunotherapy has made significant strides, challenges remain in targeting tumor cells and efficiently recruiting T cells. To address this, we developed a tumor-specific gene nanomedicine that targets a broad range of tumors and enhances T cell responses. This drug uses a specific promoter to drive tumor cells to express anti-CD3 and anti-CD86, activating T cells, and also stimulates IL-2 secretion, promoting T cell proliferation and enhancing immunotherapy effectiveness. T cell activation requires 2 signals: CD3 binding to the TCR and a co-stimulatory signal from CD86. We designed a tumor-specific gene nanomedicine that expresses anti-CD3 and anti-CD86 activators on tumor cells while inducing IL-2 secretion to promote T cell proliferation. This strategy aims to enhance immunotherapy efficacy regardless of tumor heterogeneity. We initially selected the tyrosinase promoter for its melanoma specificity, and later replaced it with the survivin promoter to target various tumor cells. 4 plasmids were designed and tested for efficacy. Nanoparticles made of PEG PLGA and DUTAP were used for nucleic acid delivery, leveraging their safety and biocompatibility. PEG improves tumor site retention and prolongs circulation, while DUTAP efficiently encapsulates nucleic acids due to its positive charge, enhancing drug delivery and selectivity. Section 2, contribution. Our project had 4 main innovation points. To propose a new therapeutic option for the treatment of tumors. Designed and successfully constructed a tumor-specific plasmid that effectively promotes T cell activation. Designed the iClan nanodrug delivery system. Successfully synthesized a novel tumor-specific expression gene nanomedicine. Immunotherapy, known for its long-lasting patient survival, faces challenges such as immune target identification, off-target effects, and resistance, often leading to high costs and limited long-term effectiveness. To address this, our team designed a tumor-specific gene nanomedicine that expresses CD3 and CD28 signaling molecules on tumor cells, aiming to activate T cell immune responses directly in vivo. Nucleic acid drugs face challenges such as limited bioavailability, degradation, and delivery issues. Traditional viral gene delivery systems, like lentiviruses, have shown success, but are hindered by risks like tumorigenicity, immunogenicity, and limited payload capacity. cationic materials also face problems, such as cytotoxicity and rapid clearance. To address these issues, we developed cationic, lipid-assisted, PEG-BPLA nanoparticles, which offer a high encapsulation rate and effectively protect and deliver nucleotides to target cells while maintaining their function. We successfully constructed these 4 tumor-specific gene nanodrugs and successfully sequenced and examined protein expression. Section 3: Engineering. Our team's inspiration was to explore whether cancer cells could be transformed into immune cells and trigger self-destruction. To accomplish this, we first focused on treating melanoma. Melanoma, a highly malignant skin tumor, has rising incidence and mortality rates, with a poor prognosis and limited treatments beyond early surgical removal. The tyrosinase promoter is an important regulatory sequence that controls the expression of the tyrosinase gene. The tyrosinase promoter regulates the transcription of the tyrosinase gene. The tyrosinase promoter is abundantly expressed only in melanoma cells, by which we can achieve tumor-specific. Induction of T-cell activation and proliferation requires 2 signals. The first activation signal delivered by the TCR-CD3 complex, and the second signal generated by a synergistic co-stimulatory molecule dominated by CD86. In vitro co-stimulation of T cells with antibodies against CD3 and CD86 to mimic the dual signaling of T cell activation has become the most widely used method for T cell activation and expansion. These 2 important activation signals are effective tools for our T cell activation. Recruitment and activation of T cells are essential for promoting immune responses. Increasing CD8 plus T cell infiltration can counteract regulatory T cell immunosuppression. IL-2 plays a key role in expanding effector T cells and NK cells, crucial for anti-tumor responses, while also supporting Tregs to maintain immune tolerance. In MHC class I deficient cancers, IL-2 can restore immune infiltration in the tumor microenvironment, enhancing therapeutic effects. Combining IL-2 with monoclonal antibodies, chemotherapy, or radiotherapy can improve outcomes. Engineered IL-2 products, like low-dose or targeted IL-2, reduce toxicity and improve safety compared to traditional high-dose IL-2 therapy. We designed tyrosinase promoter-driven NP-TRalpha CD3-CD86 mut-IL-2 to specifically express anti-CD3, scFv, and anti-CD86 on the surface of tumor cells, while mobilizing tumor cells to secrete mut-IL-2, a cytokine for T cell proliferation and survival, to activate T cells and enhance immunotherapy. We extracted plasmids from E. coli and designed primers and plasmids using SNAP gene. Finally, we constructed various plasmids using marine-derived DNA by a seamless cloning method. The constructed plasmids were sequenced, and the sequencing results were as follows. 2 plasmids were successfully constructed. Here is what we learned during this engineering. Nucleic acid drugs have limited bioavailability due to high molecular weight, negative charge, and susceptibility to degradation. iClan offers a promising solution for gene and mRNA therapy. These nanoparticles combine negatively charged biomolecules like DNA and RNA, forming stable complexes that protect them from enzymatic degradation and enhance cellular uptake, improving drug delivery through targeted mechanisms. We prepared iClan using PEG-PLGA DOTA and TAP and MC3, all FDA-approved adjuvants. PEG is biocompatible and excreted through the kidneys. TAP, a cationic lipid, is metabolized into fatty acids and glycerol, which are converted into energy via the citric acid cycle and gluconeogenic pathway. MC3, an ionizable lipid, enhances nucleic acid encapsulation and facilitates release into the cytoplasm by disrupting endosomal membranes. Here is the iClan particle size characterization we teat, and the outcomes show that the average diameter and potential self-assembly nanoparticle is reasonable and concentrated. We also test the iClan WB verification of target gene expression, and the outcomes show that alpha-CD3-CD86 proteins were successfully expressed on the cells, while mut-IL-2, a kind of secreted protein, could only be detected inside the cell, which is unexpected. IL-2 protein can be expressed with high efficiency. and mut-IL-2 can only be observed inside B16 cells, which proves the plasmid has been transcribed and translated, but mut-IL-2 is not secreted outside the cell. Next step, measure the concentration of mut-IL-2 in supernate. Here is what we learned during this engineering period. Section 4. Experiments. Here is the flowchart of our experiment. And here is our timeline. These are experiments that have been done. And this is our weekly report. And these are our experiment results. Now we come into Chapter 2, Safety. Our IGEM project develops cancer immunotherapy by using cyclic DNA from E. coli to create tumor-specific nanoparticles that activate the immune system. We prioritize safety and strict regulatory compliance throughout the research process. During the project's execution, we face potential health and safety risks associated with handling chemicals, bacteria, animals, and laboratory equipment. To address these risks, we have established detailed safety protocols that ensure each team member is equipped with the proper personal protective equipment and has undergone specialized training. We adhere to local and international regulations for safe handling and disposal of biological materials. Our safety management is guided by experienced mentors from the School of Biomedical Science and Engineering, ensuring effective risk management and quick responses to potential hazards. China's robust legal framework for laboratory safety guides are compliance with the Laboratory Safety Management Standards and Laboratory Management Regulations, ensuring a safe research environment and strict protocols for handling dangerous materials. Our laboratory management operates under a 9-tier management system, ensuring comprehensive safety oversight from the school level down to each individual laboratory. To enforce accountability, we have established clear legal responsibility guidelines, ensuring that any violations of safety protocols will be met with appropriate consequences. In conclusion, we strictly adhere to the legal framework governing laboratory safety and the school's internal safety protocols. Chapter 3, Human Practice. We have shared our synthetic biology research at regional conferences, fostering collaboration and gaining insights from fellow researchers. Cross-provincial exchanges and public outreach programs have helped raise awareness of our cancer-targeted therapies and bridge the gap between complex science and public understanding. At the Southern China Exchange Conference, we engaged in discussions with other IGEM teams, exchanging ideas on experimental challenges and solutions. This collaborative environment fostered growth, broadened our perspectives, and helped refine our project's future goals. At the Hubei IGEM meeting, we exchanged insights with other teams through presentations and Q&A sessions, focusing on experimental challenges and solutions in cancer research. We also explored commercialization strategies and concluded with lab tours, fostering connections for future collaborations in cancer research and synthetic biology. Our public outreach included school lectures to introduce students to synthetic biology and inspire interest in biotechnology careers. We held seminars in hospitals to explain our cancer-targeted therapies to patients and healthcare professionals, bridging research with practical applications. Public talks promoted health awareness and emerging healthcare technologies, fostering trust and understanding between the scientific community and the public. Collaborating with medical professionals ensured accurate and credible information in our outreach. Chapter 4, Team. Section 1, Team. We are the team from Guangzhou, China. Our lab is subordinate to South China University of Technology, Guangzhou International Campus. Under the guidance of our tutors, the team is divided into 2 groups, the wet lab group and the dry lab group. The wet lab group focuses on scientific research, handling experimental design, data collection, and analysis, while the dry lab group is responsible for publicizing the research outcomes, engaging in communication, project showcases, and expanding the project's societal impact. Our team brings together a diverse range of backgrounds, with each member contributing their unique expertise and skills to the project. Finally, we come into the attribution section. That's all our presentation. Thank you for listening.